Okay, what we're going to do now, topping off our little thing, is take a look at one family. There are a couple other kinds of things we're going to look at that are even newer and a little bit less characterized, but we're going to look at one particular class of regulatory RNAs. And what they're called is long, non-coding RNAs, or LNC RNAs. Okay, now long non-coding. Long means over 200 nucleotides long. Some are hundreds, some are even a few thousand nucleotides long. So these things can get pretty big. Okay, second thing. Non-coding means they do not provide the instructions to make proteins. They are not messenger RNAs. They're transcribed and they're used as is, or maybe processed and then used as is. Okay, now the question is, what are these things? Okay, we find that there's perhaps about 1,000 genes for these in the human genome. That's probably typical of vertebrate genes. Hundreds, probably 1,000 or more. Okay, now these kinds of things here, they're found throughout the genome. Some are in introns, some overlap with other genes. You can do that. You can have overlapping genes and two different transcription start sites. So that can sometimes happen. Sometimes you find introns, sometimes you find between genes. There's variation. Some of them are extremely conserved. But others are quite variable between different organisms. All right, now, and some of them have similarities. The sequences of some of them are similar, but not identical to that of known protein-coding genes. So it's possible that at least some of these were evolved from mutations in protein-coding genes. You may get a duplicate of a gene, and then that duplicate copy undergoes various mutations. It can no longer produce a functional protein, but the gene is still transcribed, and it may be able to take on certain other functions. Now the question is, what are the functions? We know of the functions of a few of them. And they seem to be involved, many of them seem to be involved in somehow regulating transcription. For instance, one particular LNC RNA, people using genetic engineering tricks, they deleted it. They did gene knockout of it. They wiped it out of the genome. What happened? Well, they found numerous genes were either vastly overexpressed, in other words, transcribed at far higher rates than they should be, and many others, perhaps the majority of ones that were affected, were underexpressed dramatically by factors of 10 in many cases. So it's telling you that one RNA is regulating the expression by an order of magnitude in many cases or more of a whole bunch of different genes. Okay, that's kind of intriguing. So, in theory, you could have, I'll say, 1,000 LNC RNAs. They could be regulating, in theory, or at least contributing to regulation of every single gene we've got. So that's the question. Now, a few other things about these guys. You can make fluorescently labeled nucleotide probes. Remember talking about using hybridization, or what we call in situ hybridization, actually get the stuff in the cell. You can use probes to sequences on LNC RNAs and find where in the cell, no pun intended, where in the cell are these? Couldn't resist. Okay. All right. So when you look, and you do find the nucleus, you find with certain LNC RNAs, here's the nucleus, and you find them in the nucleus, and you find that they're not evenly distributed. You find them in distinct speckled patterns. Now, if you use antibodies to proteins, label antibodies to proteins involved in transcription, various like parts of the basal complex or components of RNA polymerases and stuff like that, sometimes you find that these LNC RNAs are in the same, have the same speckled distribution as proteins involved in transcription. So that's telling you that some LNC RNAs are associated with transcription centers. We said that it appears that, in a sense, transcription occurs at discrete regions in the nucleus, where you have all the goodies together. And essentially, you move the chromatin to the center, and that's where transcription occurs. Now, the other thing is sometimes we find that these LNC RNAs co-localize, in other words, in the same speckled distribution as components of RNA processing, like small nuclear RNPs and other parts of the spliceosome. So we can see them, in some cases, in splicing centers. Once again, it appears a lot of critical nuclear functions, although we don't know nuclear substructure very well. But a lot of critical nuclear functions don't appear to be going randomly. They appear to be in discrete factories, in effect, where these things happen, and instead of proteins and other things moving down to the DNA moves towards, or that region of DNA moves towards one of these transcription or splicing regions. Okay, so we see both kinds of things, suggesting that these play a role in these kinds of processes. Now, what do they probably do? This is where we start getting more into the hypothesis direction. I think there is a little evidence for this as well. It's believed that these things function by acting as sort of scaffolds or recruiting sites for proteins involved in transcription or splicing. And possibly other RNAs as well. Now, of course, recall that proteins that are going to bind to DNA or RNA, like things that are going to be involved in transcription and splicing, nucleic acids are very negatively charged, so proteins that bind to nucleic acids tend to be highly positively charged. Well, here you have a negatively charged RNA that's going to associate in some way, take on some kind of structure, and then proteins can start sticking onto that framework. So that way, it helps to recruit and bind proteins important for these processes. And that could also explain why we see, if we get rid of them, we see transcription of a lot of genes dropping down quite a bit, because now we just don't have that framework to assemble all these proteins together. Or in some cases, transcription rising up quite a bit, because maybe transcription inhibitors need to bind to these as well as to their DNA sequences. So that's an intriguing kind of thing, and we have a lot of them. They apparently regulate transcription and possibly splicing in numerous genes. So that's one intriguing and still poorly characterized class of RNAs. Some of them being among the most highly conserved sequences in the genome, others being quite variable. Now, we can end this kind of thing about the importance of RNAs, including ones we have no idea about yet. They haven't been discovered yet. They're there, but we don't know about them yet. Okay, and regulation. Now, if you look, for instance, at a chimpanzee and humans, we're obviously quite similar in many respects. That's why baby chimps look cute, right? They practice just like baby infants do. Okay, all right, well, when you look at the DNA, they sequence the chimpanzee genome, of chimps and humans, our genomes are 98% identical. When you look at the coding sequences, the protein coding genes between chimpanzees and humans, they are over 99% identical. And although chimps and humans, and chimps are, of course, our closest biological ancestor, the divergent point between chimpanzees and humans is estimated to be about 5 to 6 million years ago. I think one of the most recent characterized hominin fossil, what they call Ardipithecus, is about 4.5 million years old. It's more on the human line than the great apes line and stuff, but it clearly has features that are intermediate, more towards human-like features, but definitely has a lot of ape-like features, and lived in kind of a mixed savanna forest environment. And that's the closest thing we have. It's not quite the ancestor of both. It's not quite the common ancestor, but it's the closest thing we have in decent fossils, although there have been fragments found even older than that. So that's an interesting kind of thing. Well, anyway, some divergence. But the thing is, there's obviously a lot of similarities, but obviously there's a huge amount of differences too. Different eating stuff. They both have similar social structures. Chimpanzee bands and human tribes are not all that different from each other in many respects. They have similar social structures and things like that, but there's significant difference. Of course, the most important thing is our brain's 3 times the size of a chimpanzee, unless you're in Congress, then it's about half the size. Okay, click resist. All right, especially since we're going at fiscal cliff in a couple more days. All right, but anyway, at any rate, very, very similar. The brain is much, much larger. Although chimps can and do use primitive tools, in fact, sometimes they even make them in advance. That's something that one was first discovered. Wow, I thought only people could do that. But, they haven't made the internet, they haven't made spacecraft, they haven't made nuclear war. It's a good thing, but considering what we do with them, they would probably be happy to nuke most of us. But anyway, they don't have the mental capacity to make a sophisticated technology like humans do. So there's a lot of differences. Even though our genomes are extremely similar and stuff, the difference that makes a man and a chimp is the expression of these genes. For instance, one particular gene that's important in neural development and is expressed mostly in the, what we call the prefrontal cortex or the frontal lobes. The expression in humans of that same gene, essentially same genes the chimp has, is many, many times greater. Genetically, those two organisms are very similar to each other. But the big difference is the expression of these genes. And what controls that is still largely a question mark. So gene sequence is just the beginning of the story. What really counts is the network of things that regulate the expression of the genes, anywhere from transcription, translation, RNA processing, whatever the case is. That's where the real story lies. And that's why we just begun to understand the details of development in molecular biology. We're just beginning to understand. We got the blueprints, but how those blueprints are used, that's a whole new ballgame. So there's a lot of cool stuff, and it appears that RNA is one of the major players in this, even though we don't know what most of them do. By the way, anybody who has any interest in this kind of stuff, I'm going to recommend a book. It's called RNA, Life's Indispensable Molecule. Now, it's more graduate level type stuff. But after this class, I think we could get, if you're interested, you can get some benefit. I was put out by Cold Spring Harbor Press last year, and the person who writes it, James Darnell, has worked in RNA biology for decades. He was also one of the co-authors of one of those big, huge 1,000-page plus molecular cell biology. That's what it's called. It's not one of the Bibles of cell biology. It's like a 1,200-page book that goes in all kinds of detail. But anyway, that is a nice review of a lot of things about RNA biology from a historical context and stuff, and he's been through almost all of it. So I highly recommend it, especially if anybody's doing papers on this and stuff. I've used it a lot. I told my wife when I was revising a bunch of things here, I said, this book is well worth every penny I spent on it. Because I used a heck of a lot in some of these notes and stuff, and a few review articles. But anyway, just gives an idea, but it does illustrate some of the interest and capabilities of RNA biology. So we see a lot of that. A lot of things about gene expression. What we've done at this point is pretty much covered key aspects of regulating gene expression. So a little bit of, for some of you who took molecular, some of this is obviously. How many people took molecular? Who took molecular here? Anyone? Oh, somebody did.
Nate?
Oh, you took, okay. Okay, I guess there's some overlap and some differences?
Yeah.
Okay. All right, cool. Okay, well, let's see what we do time-wise. Okay, half an hour. All right.